So while preparing your sample for proteomics analysis, your major concern, you're seeing all kinds of proteins, right? So you're interested in proteins. So the most important step is all the proteins of your sample should be solubilized and extracted. You should not lose any kind of protein, right? So this, what do you think? This should be included, right? This is a very important step. Second, that repetitive freezing and thawing is avoided. So, repetitive freezing and thawing, it will degrade your protein. So, it is always advised that whenever you are doing any kind of protein sample preparation or be it any other kind of biomolecule, repetitive freezing thawing cycle will always degrade the sample. So, it should always be avoided. Third is that nucleic acids are not degraded. So you are interested in proteins in your sample. So if nucleic acids are not degraded, it might hamper in your analysis for proteins. So nucleic acids are not degraded. So is it a best sample preparation technique or not? Matlab, should you degrade the protein nucleic acids in your sample or it should not be degraded? Can I have some response? Nucleic acids like DNA or RNA, if it is present in your sample preparation along with the protein of your interest, is it fine? Can you go ahead for proteomic study, or it may hamper in your protein analysis?
It is not fine. Nucleic acid presence is not fine.
Yes, correct. If nucleic acid is present, like let's say DNA or RNA, it may, especially DNA, so it may hamper your protein analysis. Okay. And fourth is that the presence of salt is maximized. This should be reversed. The salt should be minimized rather than maximized. So the best sample preparation in the best sample preparation, which options you think should be considered? First is the most, just a second. So the first and most important is the most of the proteins are solubilized and extracted. Okay, so you're performing a proteomics experiment. So your target is the whole complete proteome present in any biological sample. So most of the protein should be solubilized and you should get as many proteins as possible in your extraction process. Second is freezing and thawing. It should be avoided as I mentioned because it can degrade your proteins of interest. Okay. So these two are the steps which are very important in sample preparation. And C and D, the nucleic acids are not degraded. It should be degraded. Otherwise it will interfere with your analysis for proteins. And salt should be minimized whenever you are doing whenever you are preparing for proteomic experiment, whenever you are performing sample preparation for your proteomic experiment. Coming to uh, so this is another question related to SDS page. So SDS page is one of the simplest technology for separation of the proteins, right? So there are different reagents used in SDS page, right? So TEMID is one of that. So what is the role of TEMID? Can someone answer this? So option
Ma'am, it helps catalysis of polymerization in gel.
B, it helps in the catalysis of polymerization. Yes, very correct. TEMID helps in the catalysis of the polymerization reaction. Other option, let's see, it provides similar charge to mass ratio. This is not done by TEMET. SDS. So SDS is the detergent that is used for SDS page, and the charge to mass ratio is provided by SDS. It's a negatively charged detergent, and it provides equal charge to all the proteins of your interest. So that is why in SDS page, the separation is based just on the basis of its mass, because all the proteins now will have a similar negative charge. So B is it helps in the catalysis, correct. TEMID helps in the catalysis of polymerization. The C option was it helps in the breakage of the disulfide bonds. So this is done by reducing agents like beta mercaptoethanol and DTT. So these are used along with SDS with your proteins, so that beta mercaptoethanol and DTT, it will break the disulfide bonds, and SDS, it will denature your protein structure, as well as it will give you the charge to your protein. And cross-linking agent, it acts as a cross-linking agent. So TEMID does not act as a cross-linking agent. We use bisacrylamide as a cross-linking agent. So we are doing SDS page, polyacrylamide gel electrophoresis. So polyacrylamide itself is a gelling agent, and bisacrylamide we add to it as a cross-linking agent for preparation of SDS gel, SDS page gel. Sorry. So coming to another question. So Saina was performing refolding experiment for her protein A, and she wants to check whether protein A is folded or not. So which electrophoresis technique she should use? So options are SDS page, 2D electrophoresis, or blue native page. So can I get some response here? Which electrophoretic technique she should be performing if she wants to study a protein in a um, folded structure? So she had performed a refolding experiment. Now she want to check whether her protein of interest, protein A, is folded properly or not. And she had an idea that this protein is folded in a particular structure or whatever. So can anyone want to guess which technique should be used?
Native page.
Yes, correct. So native page, in blue native page, we are not using SDS. Rather, we are using Kumasi blue dye. So this dye does not denature the protein compared to SDS. So that is why the native structure of the protein is retained, apart from it providing the charge to the protein. So that is why whenever you are interested to study a protein, a particular protein in its three dimensional folded structure, then blue native page is a recommended technique, because in case of SDS, you will not get a folded protein structure. So suppose, let's say you have a protein dimer or homodimer protein. Okay, so it's a homodimer, meaning it is made up of two dimer, and each dimer has a molecular weight of, let's say, 40 daltons. So whenever you are doing a SDS page, so you will get a band at 40 only. But whenever you are doing a blue native page, the entire structure will be retained, and that is why you will get a band at 80, because you have two dimers. It's a homodimer, meaning of a same molecular weight and same properties. So you will get a band at 80, and you will be able to see, you will be able to separate the protein based upon its refolded form. So that is why blue native page is used for in the refolding experiment to check whether your protein of interest is folded properly or not. Now, this is another question. This is based upon a Bradford method. So Bradford method is used for protein quantification, and so you can see here when plot is given. So we'll first see the question. So Ayushi wants to perform the SDS page for 4 of her samples A to D. Okay, so she had 4 protein samples A to D, for which she first quantified the protein concentration using Bradford method of protein quantification. Okay, so she wants to quantify 4 proteins A to D, and she is using Bradford assay for her protein quantification. Okay, she had dissolved her protein samples in 50 microliter of buffer, and for each sample 2 microliter of protein sample was taken in duplicates to perform the quantification. So this is very important information. She had dissolved her protein into 50 microliter of buffer. You should note this. And of that 50 microliter, 2 microliter of protein sample was taken in duplicates, okay, to perform the quantification by Bradford assay. So here they have given a standard. Just a minute. Yeah. So, standard protein BSA, bovine serum albumin, was used to plot the standard curve, taking different amount of BSA as 2 microgram, 4, 6, 8 and 10. So you can see here the plot of this is BSA amount versus OD at 595 nanometer. So 0, 2, 4, 6, 8 and 10. So these are the different amount of BSA used, and a plot has been carried out, and at what amount, what is the OD of that particular sample is calculated, and based upon this standard curve, you have to try to find out the protein of the concentration of the protein of your interest. Okay, so given below is the standard plot obtained for BSA along with the respective absorbance. So this is the BSA amount. You can also see that from the plot, as well as here it is given. So for 0, OD1, OD2, and this is average OD. So these are the different amounts, and these are the average OD for the standard BSA. Now using this information, you have to find out what is here, the absorbance obtained for sample A to D were 0.505, 0.353, 0.184, 1845 and 0.2125. So note this down. These are the OD for the protein A to D. So from using this information, the absorbance information for all the 4 proteins and the standard GER, you have to find out first of all the concentration, the amount of the protein A to D. So now if Ayushi wishes to load 15 microgram of protein for each sample, what would be the volume required for the samples A to D? So she wants to perform SDS. So she wants 15 microgram of protein. So your target is 15 microgram of protein you should be getting. Okay. So first of all, you need to find out that what is the protein, what is the amount of protein present in your sample. Okay, that is question number 1. And second is, if she wants to load 15 microgram of protein, then what will be the volume required by Ayushi? So these are the two interlinked questions. If you answer first, only then you can answer the second question, that what will be the volume required by her. So you can take your time, solve this question, take this information, absorbance for protein A to D, standard curve, and then we can discuss this. If you have any doubt, you can ask me. So just giving you a hint, you can use this equation given to find the amount of protein A to D, okay, from this standard curve. So first calculate there using this equation. Anyone has finished at least finding the BSA amount for protein A to D? No one? So you have to use this equation. So here y, which is the OD, equals to 0.064x. X is the BSA amount. So you will get the value of x when you have the value of y, that is OD for all the four proteins. From this equation, you will get the values of x, that is the protein amount for protein A to D. Has anyone found for protein A? What is the amount? What is the amount of protein A from the absorbance given?
Protein A is 7.814 micrograms.
Correct.
Hello.
Protein. Have you found for protein B?
Hello.
Yes, yes.
Protein protein B is 5.60.
Protein B is 5.60. Okay.
Yeah.
So
Protein
how
C
have you calculated?
13.01.
Can you explain?
No, it is. I calculated in this average OD value. If the OD is 0.161 and that correspond to 2 microgram, then what will be the value for 0.505?
Okay, so depending upon this chart given, just a minute.
Yeah. వెళ్తాను మా డైరెక్ట్ ఫీల్ అవుతున్నాడు.
Okay, yeah. So from this equation, you can find the concentration, or sorry, the value of X, that is the value of protein, and from
Yeah.
that, So for also see this sentence. She had dissolved her protein sample in 50 microliter of buffer, and for each sample 2 microliter of protein sample was taken in duplicates to perform the quantification. So for this particular Bradford assay, okay, she had taken 2 microliter of protein from the 50 microliter of her protein, from the 50 microliter of her protein sample. Okay, so take this into account. Then you can find out that if she want to load 50 microgram, then what will be the volume. So here volume is given. So let's say protein, you found out value 7.8 something. So
No,
7.8.
no, no. Hello, hello. Hello,
Yes,
ma'am. Hello, ma'am. The
yes.
answer I given in the chat box, ma'am. Answer for A, 3.83 microliter. For B, 5.60. For C, 13.09.
Okay,
For D, 9.38. I already put in the chat box.
okay. Thank you. Okay, so
Yes, ma'am.
so I guess most of you would have now been able to solve. So we will just discuss. If anyone of you would have not understood, then we can discuss it. So this is the calculation. So from this equation, okay, first you need to calculate the amount of protein. Okay, now see what is given for 50 microgram protein. What is the volume required? Okay, so now it is given that 7.9 microgram for 2 microliter of protein, because you are using 2 microliter of protein for your Bradford assay. Okay, so for 2 microliter of protein, you got this. This is the amount, 7.8 or 7.9, something around that. So this is the amount of protein. So now you want 15 microgram of your protein. Okay. If you want to load 15 microgram of protein, then what? Uh, yeah. So if you want to load 15 microgram, then how much volume you will require? So it is simple mathematics. 7.9 microgram for 2 microliter. Then for 15 microgram, how much? So you will get around this much, 3.9 microliter. So for 15 microgram of protein to be loaded, you need to use 3.9 microliters. And similarly, you can calculate for other proteins. Okay. So I hope this is clear. If anyone has any doubt, they can ask. Okay, nice. I think everyone has understood. So it is nice. Great. Someone wants to join. So we will move now to another question. So this is another question. Saima works on meningioma cell line samples, and she wants to do proteomic analysis taking cells from patient derived cell lines. So which of the following methods she should use for cell disruption during sample preparation? Sonication, B, manual grinding, C, freeze thaw, and option D is all of these steps she should be using for her sample preparation. So, what do you think? What is the method for cell disruption? What kind of method should be used? Can I get some response?
Freeze-thaw.
Freeze thaw. But we already discussed in the earlier question that freeze thaw will affect your proteins, and it might cause degradation of your proteins. So that is repetitive freeze thaw we should avoid. So in any way we should avoid freeze thawing. Then how can we use that for cell disruption?
Sonication.
Yes, correct. Sonication. So sonication helps in cell disruption during the sample preparation. Okay. So generally, whenever you are doing Hello?
వెళ్తాను రాడు బయట కేట్టుకున్నాడు
I'm not
ఫోటో
able to
పెట్టండి
hear you.
పెడతాను విషయం లేదంటే ప్రాజెక్ట్ విషయం లేదు మన దానిలో ఉంది విషయం ఇది తిరిగినా పెద్ద టైం పడుతుంది
If you're saying something, you are not audible properly.
ప్రాజెక్టులు ఉన్నాయి మన దగ్గర
Janaki, you can mute yourself.
ఎస్ఎంఆర్ జేఆర్ఎఫ్ అప్లై చేశారు
Janaki, Ramya, you can mute yourself. Yeah, thank you.
Okay, we'll move to the another question now. So, which of the following technique is not used for removing salt from a proteomic sample? So, following are the options given.
గ్యాస్
Dialysis,
క్రోమాటోగ్రఫీ
sorry,
గ్యాస్ క్రోమాటోగ్రఫీ
gas chromatography. You use that for salt removal.
నాట్
Correct, correct.
నాట్ ఫర్ సాల్ట్ రిమూవల్
Yes, yes, correct. You use dialysis, spin dialysis, and gel filtration. So, definitely, gas chromatography is the correct answer. You don't use it for salt removal. Salt removal. So, this is another question. Triazole method is used for the extraction of the which of the following biomolecules? Proteins and RNA, proteins and lipids, DNA and RNA, or proteins, RNA, and DNA? Anyone wants to answer for triazole method? What kind of biomolecules you can extract using triazole method? Any guesses? Am I audible?
Option D.
Yes, you are audible.
Option D, yes, correct. Proteins, RNA, and DNA. So, this is a very unique method where you can try to, from a single sample, you can extract different kinds of biomolecules, protein, RNA, as well as DNA. So, so in triazole method, it is basically used for RNA, but you can also separate proteins and DNA by this method. So this is the depiction of sample using triazole reagent. So Vipin wishes to extract RNA using triazole method. During the extraction, following three distinct layers appears in the tube. Which of the following layers he should further use for extracting RNA? So uh, Vipin has treated his sample with triazole reagent and then you used other solvents like chloroform and then performed the centrifugation and uh, he has been he showed three separate layers. Okay, layer A, layer B, and layer C. So, which is the layer where RNA is present? Can you comment upon that? So, layer A, layer B, or layer C? Or you think that RNA cannot be extracted? But we already discussed, right, in the earlier previous slide that triazole method is used for all the three kinds of biomolecules proteins, RNA, as well as DNA. So, in which okay, someone has joined. So in which layer RNA is present?
Top layer.
What? I was not able to hear. Top layer.
Layer A.
Layer A, correct. Layer A. So RNA is present in the top aqueous layer. In the layer, just a minute. So here your DNA will be present, and in finally the organic layer, the last layer down, the proteins will be present. So this is a very unique and useful method because from a one single sample, so let's say you are working on some human serum sample or plasma samples which are very restricted. I mean, you are not able to get so many samples and you want to perform uh, RNA, DNA as well as protein analysis. You want to do proteomics, you want to perform genomics as well as you want to also study transcriptomics. So this is a very nice technique because here you can right from a one sample, you are taking one single sample and you can extract three different kinds of biomolecules. So trizol is very unique in this sense that you can target different kinds of biomolecules by a single method. And in so, triazole basically contains guanidinium thiocyanate. Okay, so that helps in it inhibits the RNAs, and as you know that RNA is very labile. Uh, it gets easily degraded because of the RNAs, and RNAs is present in very freely in the environment. So that is why whenever you are performing mRNA or you are working with RNA extraction, you have to take extra precaution. You have to clean everything with RNAs free water so that your RNA is not degraded. So triazole is a nice method for RNA extraction, but along with it, you can also extract DNA and protein. So we will move to the next question. So, which of the following holds true for one-dimensional SDS page and two-dimensional electrophoresis? Option 1 is unlike a 1D SDS page, staking gel is not required for 2DE. Second option is like 1D SDS page, isoelectric focusing is performed in 2D also. Third is in 1D SDS page, proteins are separated on the basis of molecular weight, whereas in 2D SDS page, proteins are separated on the basis of mass to density ratio. TEMID is not required for casting gel of 2D, which is required for 1D SDS page. So, which is the correct statement amongst the four statements? Any guesses? So, TEMID, we have already discussed in earlier question, right? So, TEMID is used for as a catalyzing agent uh, during the polymerization reaction of page. So TEMID is not required for casting gel for 2D, which is required for 1D. So if you are making gel for 2D also, you are using SDS page in the second dimension, you will require TEMID or not? What do you think? Here also you are using SDS page, but in two-dimensional, first you separate the protein based upon isoelectric focusing. So that is your one dimension. Once it is done, then again, like SDS page, you are separating based upon its molecular weight. So it is the same like SDS page. Okay. In the second dimension, you are separating depending upon its molecular weight. So you will require all the reagents which were you were using for one dimensional SDS page also. Okay, so that is why this is not a correct option. TEMED is not required for casting gel for 2D, which is required for 1D. Okay, so it is required as well as in 2D. 2D is 2D means what you are separating using two dimensions. So first is isoelectric focusing and second is SDS page. While in one dimensional SDS page, you are just separating based upon SDS page. So because even in 2D you are using SDS page, that is why TEMID will be required. Now we will look at another option. So this is not the correct. This does not holds true. Now we'll go to option C. In 1D SDS page, proteins are separated on the basis of molecular weight. Proteins are separated on the basis of molecular weight, correct? Whereas in 2DE, proteins are separated based on the basis of mass to density ratios. So are we separating in two dimension based upon mass to density ratio? Can I have some response here? So I have already mentioned that in two dimensional you are using isoelectric focusing, okay, and another is the SDS page. So that is the two dimension. So it is not based upon mass to density ratio. So even this is not correct. So B, like 1D SDS page, isoelectric focusing is performed in 2DE also. So in 2DE, you are performing isoelectric focusing, but in 1D, you are not using isoelectric focusing. You are just using SDS page for your separation. So this is an incorrect part. So even this statement is not correct. Now coming to option A, unlike one-dimensional SDS page, stacking gel is not required for 2D, which is correct. You only require resolving gel in case of two-dimensional electrophoresis. Okay. So this there comes the ends to this question answer session. So if you have any doubt in any question that we have discussed in this session or even in the previous session or while going through any videos or lectures, you can ask now, or you can also write send you can also mention that in Google Sheet that we upload. Any queries? Am I audible?
Yes, Neha, you are.
So if no one has any doubt, I will stop sharing my screen.
Uh, Sneha,
Ma'am,
someone is asking, can they have the last session recording video?
Yes, so I have shared it on YouTube. Yeah, sure,
Uh,
I can give that recording.
so,
So
Sneha, you can you can post the same YouTube link in the announcement so that everyone can access.
okay, okay, okay,
Yeah.
sure. So yeah, I have the recordings. I'll share that also. Okay, so I hope you had enjoyed the session and hope to see you for the next session. Thank you very much, all of you. And I'm ending the session now.